Okay, today we will discuss about the enzyme. Okay, and this is the first lecture of the enzyme. And before we start this session, I want to say something about the enzyme. What is actually enzyme? As you know that in our body or in different living organisms, as you know that in our world, in our earth, there is presence of so many living organisms, and in that living organism there is occurrence of so many functions, so many procedures, so many functions, so many activities that are going on day by day. Okay, but if you see this reaction or this process which are occurring inside the body, that will took more time if there will be no enzyme. Okay, enzyme is a substance, or this is a protein in nature. Okay, when we see the enzyme, that is chemically proteinous in nature, and that protein actually do what? That proteinous substance, it accelerate it accelerate the, or it increase the rate of function, rate of chemical reaction in the body. Okay, in our body or in every living organism, there is occurrence of several procedure. Okay, and these procedure which are, or the process which are occurring in our body, that procedures that become maximum, or that process that become faster due to the presence of one of the substance that is called as enzyme. So enzyme do what? Enzyme, it is a substance which increase the rate of the reaction that taking place in the body, that is taking place in the all living organism. Suppose in case of our body that performs so many functions, just like your, in our body there is occurrence of your digestion process. Okay, in the digestion process there will be requirement of the enzymes, and the enzymes that required during the time of your digestion, that are called as your digestive enzyme. And I think you all study about the DNA replication. DNA replication is a process that is taking place in our cells, and for the DNA replication there will be requirement of some enzyme. Okay, to complete the DNA replication process there will be requirement of the enzyme. If there will be no enzyme, then that procedure will be very slower, then that process will very slow, so that that will not complete in a particular time. Okay, as you know that digestion should occur in within some very few time, so that we are able to digest that food, or that digested food can be utilized in our body organization. Otherwise, if it is not able to digest by our body, then it will not able to utilize in our organization, our body organization. Okay, so every day we are requiring the enzyme in our body. and everything that is entering into our body, that is digested by the presence of the enzyme, that is called as a digestive enzyme. Okay, so enzyme means, actually we will discuss its historical background, and then we can find that what is the meaning of that en, and there is another word, that is your zyme, enzyme. Okay, and enzyme, that is means that is inside the yeast. Actually that was first discovered from the yeast, so that is called as your enzyme. We will discuss that discovery. So simply, enzyme means what? Simply that are one kind of catalyst. In the chemistry, you can you may study about the catalyst. What is the function of the catalyst? The function of the catalyst is that it will accelerate the function, or it will accelerate the reaction, so that the reactant will convert it into a product in very small period of time. The reactant will convert it into a product in very small period of time. For that there will be requirement of what? For that there will be requirement of catalyst. Like that, in our body or in living organism, there is occurrence of several process, and that process will accelerate, or that process will increase, or the rate of that reaction will increase when there will be presence of what? When there will be presence of your enzyme. So enzyme are also called as your biocatalyst or biological catalyst. Okay, in the chemistry, the catalyst increase the rate of reaction, so that in very small period of time the product will convert, the reactant will convert it into your product. Like that, in our living organism also, in the living system also, there is presence of the biological catalyst, and that biological catalyst that increase the rate of your process or rate of the reaction, so that in very small period of time we will get the product. Okay, so that is your enzyme. Okay, so here in this class we can study its structure, okay, and its classification and its nomenclature. So let's come to the next slide. Okay, so enzyme are called as what? These are called as your biological catalyst. What is the meaning of biological catalyst? These are the catalyst, or catalyst means the substance that, catalyst means what? The substance that increase the rate of reaction. Okay, the catalyst means the substance that increase the rate of reaction, that is called as catalyst. And these enzymes are the organic. Enzymes are what? They are organic, because this enzyme that is the part of our biomolecule. This is a biomolecule. Okay, and basically the enzymes, every enzymes that are proteinaceous in nature, except some of the enzyme that is called as a riboenzyme. Okay, or some of the RNA that act as enzyme also. Some of the RNA that act as enzyme also. So except that RNA, but all other enzymes which are present in a cell or in living organism, all of the enzymes are proteinaceous in nature. And as you know that protein is a what? Protein is a biomolecules. So enzymes are also organic. Thermolabile means these are if we provide thermal heat. Okay, if we provide the heat, then the enzyme will not able to function. If we increase the rate, if we increase the temperature, then what will happen? The function of the enzyme that will disturb. The function of the enzyme will disturb. So that is called as your thermolabile, not a thermostable. This is not stable in the high heat. If we increase the heat more than your 55 degrees Celsius, then enzymes stop working. Okay, so that is called as what? Thermolabile catalyst. Okay, and that increase the chemical reaction without any changes. Okay, without any changes, if there will be presence of your enzyme, it will not change anything. Only it has to increase its reaction process, or it has to increase its rate of reaction. Okay, but there will be no change in the product or in the reactant. So they accelerate the rate of chemical reaction. They accelerate means they just increase its rate, or the process that will become speed up. The process will become speed up, and they accelerate the rate of chemical reaction without being consumed in the reaction. In the reaction, the enzyme will not consumed, or it will not be utilized. Okay, it is not included in the reaction. After its function, it will remove. After its function, it will remove. It will just give the acceleration, or it will just increase its rate of reaction. it will not include or it will not consume in the reaction. Okay, so let's see here. Enzyme, they are very important protein. Okay, and that are present in our body, and this is another part of our biomolecule. Okay, and when we see its history, okay, when we see its history, it was first discovered by the scientist, that is Edward Buchner. It was first discovered by the scientist Edward Buchner, and this discovery was actually discovery by chance. Discovery by chance. What happened? One of the experiment that was done by the Edward Buchner. Edward Buchner was studying on yeast. Actually, he was working on yeast, and he grew some yeast on sucrose solution. He took some sucrose solution, and in that sucrose solution, he grew some yeast. Okay, and after few days, he found that the sucrose has got changed. Okay, what happened? After a few days, he found that the sucrose has got changed. From that, he confirmed that yeast has some substance, okay, in it, and which is causing changes in the sucrose. What happened actually? Actually, what happened when he put the yeast in the sucrose solution, after a few days, the sucrose converted into alcohol. The sucrose converted into alcohol. Okay, when you put some yeast here, okay, when you put some yeast and with this sucrose solution, after a few days, what happened? The sucrose is converted into your alcohol. From that, your Buchner, he confirmed that yeast has something in its cell. In the cell of the yeast, there is present something, and that something or that substance is going to change this sucrose into alcohol. Okay, and the term from that, it was given as the term enzyme, because the first time that substance which is discovered by your Edward Buchner, that was actually enzyme. But how this name was given? How this name was given? Okay, let me clear. The term enzyme was given by Kuhne. The term enzyme was given by Kuhne. But what is the meaning? What is the literal meaning of this enzyme? If you see En, and means in. En means what? In. And zyme. Zyme means that is your yeast. Because the substance which just convert this sucrose into alcohol, that is actually some substance, and that substance was named as enzyme because that substance was first discovered by or discovered from the yeast, as it was first isolated from the yeast, so that is called as your enzyme. En means in, and zyme means what? Zyme means yeast. Okay, so something is present in the yeast, and that something means that is now called as your enzyme. That something is now called as what? Enzyme, and that enzyme convert this sucrose into alcohol in very few period of time. Okay, so this was the historical background how this the term enzyme was given. Enzyme means in the yeast, as the enzyme or the substance that is present in the yeast was discovered first. That's why that is called as your enzyme. Okay, and the scientist James Sumner, who found that enzymes are proteinaceous in nature. Actually, all the enzymes except one of the enzymes, that is your riboenzyme, except ribozyme, all the enzymes are what? All the enzymes are proteinaceous in nature. All the enzymes are what? They are proteinaceous in nature. They are made up of what? Proteins. And as you know that proteins are made up of what? Proteins are made up of your amino acids. That means the enzyme are also having what? The enzyme are also having the amino acids. Okay, let's see. All enzymes are protein in nature, that I have discussed earlier, but except one thing, except one enzyme, that is called as a ribozyme or riboenzyme. And the riboenzyme means these are RNA in nature. Riboenzymes are RNA in nature. Okay, some of the RNA that act as catalyst. Okay, there is something there is presence of some RNA that act as catalyst. Okay, for example, in case of your protein synthesis, during the time of protein synthesis, the ribosome have rRNA. In the ribosome, there is presence of rRNA, and for the initiation of protein synthesis, there is a requirement of a sequence. That sequence is called as a Sine-Delgarno sequence. When we discuss that translation in upcoming classes, I will clarify that Sine-Delgarno sequence. In the RNA, actually in the mRNA there is presence of a sequence that is called as your Sine-Delgarno sequence, and in the ribosome, there is presence of what? Ribosome represents this, there is presence of your RNA, and that RNA having a anti-Sine-Delgarno sequence, and that sequence is your AGG-AGGU. Let me make here. Suppose this is the mRNA. In the mRNA, there is presence of a sequence that is called as your AGG-AGGU. This is called as your Sine-Delgarno sequence. This is called as Sine-Delgarno sequence. Okay. And in the ribosome, there is presence of RNA, and in that RNA, there is presence of anti-Sine-Delgarno sequence, that is UCC-UCCG. Sorry, UCC-UCCA. Okay, this is called as your anti-Sine-Delgarno sequence. Anti-Sine-Delgarno sequence. Okay. So this is the rRNA. This is the rRNA. This RNA that bind with this Sine-Delgarno sequence of the mRNA. This is the mRNA. Okay. This is the ribosomal RNA. This is present inside the ribosome. This is present over the mRNA. When this Sine-Delgarno sequence and anti-Sine-Delgarno sequence, they will bind, here the ribosome will start to bind in this mRNA. And for the process of your protein synthesis, there will be a requirement of attachment of ribosome. So for the initiation of protein synthesis, there will be requirement of attachment of ribosome, and that is possible due to the presence of the RNA inside the ribosome, and that kind of RNA that are using or that are helping in the initiation of protein synthesis. So that they are also involved in the reaction process. One kind of reaction, that is your translation reaction process. So that kind of RNA, if they are using as an enzyme, that are called as your ribozyme. But this enzyme or this rRNA, that is not proteinaceous in nature, that is RNA in nature. But except this RNA enzyme, or except this riboenzyme, other enzymes which are present in the earth or present in the world or living system, that are proteinaceous in nature. We have to fix this in our mind. Every enzyme that is present in the nature, that are proteinaceous in nature, except our ribosome. Okay. Okay. What is the difference between the protein and enzyme? Okay, from this diagram we can clear that. What is the difference between protein and enzyme? Okay, so this whole red part, that is your protein, and a small part of that protein, that is our what? That is our enzyme, and some of the enzyme that are RNA also. So this diagram, very beautifully, this diagram representing the relationship between the protein, enzyme, and RNA that act as your enzyme. Okay, you can see here some of the protein. Okay, here this is the some part of your protein. Some of the protein that are enzyme in nature. Some of the protein that are enzyme in nature. That means all the proteins are enzyme. All the enzymes are proteinaceous in nature. Here all the proteins are all the enzymes are proteinaceous in nature, and some part of the protein that are also some part of your enzyme that are also what? That are also RNA. This is the representation. How can we differentiate between the protein, enzyme, and RNA that may act as your enzyme? So all enzymes are protein except some RNAs. Okay, except some RNA. All enzymes are protein, but not all the proteins are enzyme. Not all the proteins, these are not included in your enzyme. That means not all the proteins are enzyme. Some of the enzyme or all the enzymes that are proteinaceous in nature, except what? Except the RNA. But RNA ke chhadi dele baki jitki rahela, all these are what? All these are enzyme. Okay, and these enzymes, all every enzyme that are proteinaceous in nature. That means that are made up of protein. Okay, so when we see enzymes, then the enzyme are classified into two types. Actually, whole enzyme that is classified into your two types. One is your simple enzyme, and another one that is the conjugative enzyme. Okay, one is the simple enzyme, and another one that is the conjugative enzyme. What is the meaning of simple enzyme and conjugative enzyme? When we see, if whole enzyme is made up of protein only, if it is made up of protein only, if any enzyme that are having only protein, then that are called as your simple enzyme. Okay, simply if the enzyme is only made up of what? If it is only made up of your protein, then that is called as your simple enzyme. Make this in your mind. And conjugative enzyme means if it is made up of protein and it is also having non-protein part. It is not only made up of protein, it is having some kind of non-protein substance also. That is called as your conjugative enzyme. So in the conjugative enzyme, there will be presence of your two parts. One is your protein part and another one that is your non-protein part. Clear? And the protein part in the conjugative enzyme is called as your apoenzyme. Okay. See here, enzyme that is classified into two types, whether there is presence of only protein or with protein there is presence of some non-proteinous substance. Okay, if there is only protein, that is called as a simple protein. If there is presence of your protein with non-protein substance, then that is called as your conjugative protein or conjugative enzyme. Okay, so in the conjugative enzyme, there will be presence of your protein part and non-protein part. The protein part is known as what? That is called as your apoenzyme, and the non-protein part is called as your cofactor. Okay, here in case of your conjugative enzyme, there is presence of two parts. The protein part is called as your apoenzyme, and the non-protein part is called as what? Cofactor. Okay, so apoenzyme, or this is also called as apoprotein. Simply we may write them as apoprotein. Okay, this is also called as what? Apoprotein. Okay, so in the conjugative enzyme, this apoenzyme and cofactor, they will get joined. Suppose this is your apoenzyme, or this is the protein part. When the cofactor will bind, then this is the complete enzyme. Now this is the complete enzyme. Without cofactor, it will not able to perform its function. So when both apoenzyme and cofactor they will bind, they are making a structure, or that enzyme is now called as what? Holoenzyme. What is holoenzyme? When the protein part and non-protein part, or the apoenzyme and cofactor, they will get bind or they will get joined, that are making the enzyme that is called as what? Holoenzyme. Okay, so in the holoenzyme there is presence of apoenzyme and cofactor. Okay, apoenzyme and cofactor, they are making the holoenzyme. Holo means what? Holo means whole. It is coming from the word whole. Okay, whole enzyme is now having your protein part and non-protein part. The protein part, that is your apoenzyme, and the non-protein part is called as your cofactor. So I have made some graphical representation of the relationship between your apoenzyme and cofactor. This is the apoenzyme. This is only having your protein. This is only having the what? It is only having your protein. And this is the cofactor. This is not proteinaceous in nature. This is not protein. Okay, it may be organic substance, it may be inorganic substance. Okay, it may be a organic substance or it may be a inorganic substance. So here, when the enzyme and the cofactor, this is the cofactor, this is the enzyme, when both get joined here, apoenzyme, okay, this is the apoenzyme and this is your cofactor, when they get joined here, now that is called your holoenzyme. Okay, this is a small representation of your enzyme and their relationship with the cofactor. Okay, one of the question is here, what is the relationship or what is the difference between the biocatalyst and blastula? Blastula, actually, that is the part of your embryonic development in case of your human being, but here we are discussing about the biocatalyst. And biocatalyst means what? Biocatalyst means some of the substance that increase the rate of reaction, that are called as your biocatalyst. Okay, some of the substance that are living substance, or the biomolecules, that are some biomolecules, that biomolecule increase the rate of reaction, that are called as your biocatalyst. But blastula, that is one kind of that is the part of your animal reproduction, human reproduction or animal reproduction. There is a stage at which there will be presence of mass of tissue or mass of cells, that is the embryonic cells, and that is called as your blastula. So there is so many differences between your blastula and biocatalyst. There is no relationship between blastula and biocatalyst. Okay, anyway, this is not in this this is not the part of this topic. So let's see here, enzyme and cofactor. This is the relation, another relation I'm making here, enzyme and your cofactor. When we see the enzyme, this is the enzyme. Okay, this is the enzyme and this is the cofactor. What is the function of this cofactor actually? Let's see here. And this is the substrate. What is the meaning of substrate? We will discuss later. Anyway, let me make you clear that. As you know that during the time of your reaction, there will be requirement of what? There will be requirement of your reactants, and this substrate, that is your reactant. The substrate, that is the reactant. So this reactant will bind with the enzyme. The substrate will bind with the enzyme at a specific site. Okay, this substrate will bind with the enzyme at a specific site, that is called as your active site. And that active site, when at the active site the substrate will bind, then it will just convert this substrate into product. So for the specificity, to maintain its specificity, to get proper joining of that substrate, there will be requirement of your cofactor. Proper functioning, for the proper functioning, there will be requirement of what? There will be requirement of your cofactor. So that, let's see here, this is the cofactor. This blue part is the cofactor. This blue part is your cofactor, and here this is the substrate. When if there is presence of your substrate here, this substrate cannot bind here. Okay, before we go to the upcoming part, let me clear something here. When the substrate come, then enzyme also come. The enzyme has to bind with the substrate. Okay, and there should be a proper orientation, or properly that substrate will bind with the enzyme, then only the enzyme can function. Okay, if enzyme is not properly binding here, suppose this is the enzyme, here there is no proper space for the binding of your enzyme. There is no proper space for the binding of the enzyme. So this reaction will not possible here. So that what happened here, the cofactor come to bind here. Here the cofactor come to bind here, and now the substrate will comes to bind with this cofactor. That means the cofactor gives the functionality to the reaction. Okay, or that enzyme will able to function properly. So that functionality to the enzyme, that is given by the cofactor. Okay, for the proper functioning, the cofactor is there. Clear? So this is the cofactor. Then when we see the cofactor, there are two kind of cofactor. One is your organic, or that may be your inorganic. The cofactor that may be organic, or that may be your inorganic. So what if we see the organic cofactor? The organic cofactor are very small and having low molecular weight, and they will bind with the apozyme. Yes, cofactor will bind with the apozyme. Let's see here. This is the apozyme. Just wait. This is the apozyme. Okay, this is your what? This is the apozyme or apoenzyme. Okay, this is the apoenzyme, and apoenzyme and cofactor will bind, and they will make your enzyme. Okay, this whole structure, that is your that is the complete enzyme. Okay, because it will not able to perform its function without the cofactor here. Okay, so let's see here. So the apozyme will bind with the organic cofactor, or it may bind with the inorganic cofactor. So when the apoenzyme, if you see the apoenzyme, then there is presence of, when we see the cofactor, organic cofactor, then there is two kind of organic cofactor. One is your coenzyme and another one is your prosthetic group. Okay, let's see here. The cofactor is again divided into two types. One is your coenzyme, another is your prosthetic group. Okay, first I have to make you clear that the apoenzyme having your apoenzyme will require what? Cofactor for its functionality. So cofactor may be organic or inorganic, and if it is organic, then one kind of organic cofactor, that is your coenzyme, and another one, that is the prosthetic group. The coenzyme and prosthetic group, what is the difference between your coenzyme and prosthetic group? What happened actually? The coenzyme means this is the cofactor, it will not bind tightly with the enzyme. Okay, when we see, sometimes what happened, the cofactor will not bind tightly. Okay, and sometimes what happened, the coenzyme bind tightly. Sometimes it will not bind with the apoenzyme tightly, and sometimes it can bind with the apoenzyme very tightly. So if it is not bind very tightly, not, or it is loosely bind. It is loosely bind with apoenzyme. That is your coenzyme. Okay, it cooperates the enzyme to make its function. Okay, and prosthetic group. If the cofactor is prosthetic group, that is also organic substance or organic compound, but it will bind tightly. It will bind tightly with the apoenzyme. Okay, with the apoenzyme, it will bind very tightly, whereas your prosthetic group, that will not bind tightly with the apoenzyme. That is the basic difference. Okay, we will take some example, then we will clear that. Then inorganic, that may be your metal or that may be your non-metal ion. Okay, so if it is metal, then metallozyme, that is also called as a metallozyme. Anyway, let's come to the coenzyme. Okay, let's see here. Cofactor is divided into two types. One is your organic, another is your inorganic. Then the organic is again divided into coenzyme and prosthetic group. Coenzyme, that is loosely bind with the apoenzyme, and the prosthetic group will bind tightly with the apoenzyme. So let's discuss more thing about your coenzyme. What is coenzyme? They are loosely bound with the apoenzyme. Yes, I have talked this earlier. And it may detach from the apoenzyme. Sometimes what happened? It may detach from the apoenzyme. Okay, if it will detach, means it will remove from the as it is not tightly bind with the apoenzyme, it may remove out from the apoenzyme, and it may join with the another enzyme also. It may join with the another apoenzyme also, so that it may detach from the apoenzyme. One of the example of your coenzyme, that is vitamin C, and the chemical property of your vitamin C, that is your ascorbic acid. Okay, what is the role of this ascorbic acid? Let's see here. The ascorbic acid that will bind with the enzyme, that is the apoenzyme. This is the apoenzyme. Okay, it will bind with the apoenzyme. Which apoenzyme? That is prolyl hydroxylase. That is prolyl hydroxylase. That is the enzyme. What is the role of that prolyl hydroxylase? When the prolyl hydroxylase that bind with the vitamin C, the vitamin C, that is your cofactor. Okay, or that cofactor is your coenzyme. This is your cofactor, and that cofactor is the coenzyme. That means it will not bind tightly with this prolyl hydroxylase enzyme. Okay, and what happened? If the vitamin C bind with this prolyl hydroxylase, it helps in the formation of collagen. Collagen, what? That is a protein. Collagen fiber. Okay, collagen fiber that is present everywhere in our body, and that is a proteinaceous structure, and that make the skin. Suppose this is the skin. The skin is elastic. The skin is elastic due to the presence of a fibrous protein that is called as your collagen fiber protein. Okay, and that collagen fiber protein that is formed when we are getting some injury in our skin. If in our skin there is occurrence of any injury, then that injury will remove, or that injury will get relieved when there will be a formation of your collagen fiber. When there will be formation of your collagen fiber in that injured part, then that will complete here. Okay, or that is called as wound healing. The wound healing means kshata. When the wound healing is occurring, there will be formation of your collagen. Okay, you will maintain again. So during wound healing, the collagen form. Okay, and if the vitamin C is lower, if the vitamins in our body is lower, the wound healing is not possible, because for the wound healing there will be requirement of your prolyl hydroxylase enzyme. That prolyl hydroxylase enzyme that is required to form your collagen fiber, and that collagen fiber make the skin elastic in nature. Okay, that make the skin elastic, so that during wound healing or during injury healing, the collagen that will form. And if the vitamin C is lower or the vitamin C is not present, then it will take more time. The wound healing will take more time. The recovery of that injured part will take more time if there is presence of your lower level of vitamin C. And if there is presence of higher level of vitamin C, then vitamin C will bind with the prolyl hydroxylase enzyme. So the prolyl hydroxylase enzyme will work properly in presence of your vitamin C, or it will not work properly in absence of your vitamin C. Okay, but your enzyme, that is the apoenzyme, that is your prolyl hydroxylase, but this vitamin C, that is not proteinaceous in nature, or that is not your enzyme. But if that is present, then the process will become success, or the process will become more faster. Okay, so that is the role of your coenzyme. Okay, but here the coenzyme will bind loosely with the apoenzyme. Okay, another one, that is the prosthetic group, and that is also organic cofactor. And apoenzyme and prosthetic group, they will bind properly or bind tightly. The prosthetic group will bind tightly with the apoenzyme. And here one of the example, heme group in the hemoglobin. When we see the hemoglobin that is having your 4 polypeptide chain, the hemoglobin that is having your 4 polypeptide chains, and in that polypeptide chain there is presence of a structure that is called as your heme group, and that heme group is connected with our iron. That heme group is connected with the iron, and that is tightly bind with the, that heme group tightly bind with the RNA in case of iron in case of a hemoglobin. So that is, if any enzyme that is having prosthetic group, then that prosthetic group will bind tightly with that apoenzyme, so that it will not remove easily from that apoenzyme, or for the removal of that there will be requirement of high energy. Okay, so that is called as your prosthetic group. Okay, another one, that is your inorganic cofactor. The organic cofactor is divided into 2 types. One is your coenzyme, another one is your prosthetic group. The coenzyme loosely bind, the prosthetic group tightly bind. Whereas when we see the inorganic cofactor, it may be metal or non-metal ion. The inorganic cofactor, there may be your metal or non-metal ion, and they may loosely or tightly bind with the apoenzyme. Okay, so one of the example, that is your phosphorylase enzyme. Phosphorylase enzyme, sorry, this is phosphorylase enzyme. One of the enzyme, that is phosphorylase enzyme. Okay, the phosphorylase enzyme, that phosphorylase enzyme use the magnesium ion for its activity. Means the magnesium ion act as cofactor. Okay, suppose this is the apoenzyme. Okay, then here there will be attachment of your magnesium ion, so that the apoenzyme will become functional. Okay, and if enzyme require metal activator, so this is called as your activator. We may say them as activator. If there will be requirement of your metal activator, then that kind of your enzyme is called as your metallozyme. If metal is required for its functionality, then that kind of enzyme is called as your metallozyme. Okay, one of the example, most useful example, that is your lipase. What is the function of lipase? The lipase will break the lipid. The lipase will break the lipid, and for the functioning of that lipase enzyme, there will be requirement of ion, that is your calcium ion. There will be requirement of the calcium ion. So that this is your inorganic cofactor. They will bind with the apoenzyme, so that the apoenzyme will become functional. That is called your inorganic cofactor. Okay, so let's see some terminology. So before that, we are going to this terminology. We have to fix this in our mind, that enzyme. First of all, we have to know the enzyme. There are 2 kinds of enzyme. One is your simple enzyme, another is your conjugated enzyme. In the simple enzyme, there will be only presence of your protein part. But in case of your complex enzyme or conjugative enzyme, there will be presence of your one protein part and another non-protein part. The protein part, that is called as your apozyme, and the non-protein part, that is called as what? The non-protein part is our cofactor. The cofactor again of 2 types. One is your, whether that is made up of your organic substance or inorganic substance. If it is made up of your organic substance, then that is what? Then that is your coenzyme. Okay, or that may again of another type, that is your prosthetic group. If it is inorganic, then it may be metal ion or non-metal ion. If it is metal, then that is called as your metallozyme. That is called as your metallozyme. So cofactor, that helping in the functionality, to maintain the functionality of the enzyme, there will be requirement of your cofactor. Anyway, let's come to the basic things about your enzyme. Okay, basic thing. we have to know about the enzyme. So some terminology or some term we have to know before we go to the maximum part of our enzyme. Let me clear what are some terms that is associated with the function of your enzyme. One of that word, that is your substrate. Substrate means what? It is the reactant. Substrate means what? It is the reactant in the reaction. In the chemical reaction also there is a requirement of your reactant. That reactant will convert it into what? Product. The reactant will convert it into product. And for that process of conversion of reactant into product, for the conversion of reactant into product, in a faster way, there will be requirement of the enzyme. So anyway, we are not going to that part. First of all, let me clear what is substrate. This is the reactant which bind with the enzyme. Okay, let me write the reaction here. The reactant will convert it into your product, but it will take more time. Okay, simple. If you see this simple reaction, then it will take more time. It will take more time. But when we put the reactant with the enzyme, then the product will convert in very faster time. Okay, so it will took less time. Okay, it will took more time, but it will took less time. In very few time, there will be, or very few second, that will convert it into your product. Clear? So enzyme, when bind with the substrate, the enzyme has to bind with the substrate, so that the enzyme will bind with this reactant. Then there will be conversion of your product. So first of all, what happens? The enzyme will bind with the substrate, and it will make the enzyme substrate complex, and that enzyme substrate complex is temporary. Okay, after the conversion of your product, that enzyme will dissociate from that substrate. The enzyme will dissociate from the substrate. Then what is product? That is the end result of the reaction. We know that that is the end result of the reaction. And another things we have to remember, that is the active site. What is active site? The part of the enzyme which will involve in the reaction process. Okay, the part of the enzyme or enzyme's catalytic site, the part of the enzyme which increase the rate of reaction, the site or the area of the enzyme which is catalytic in nature, that is called as what? Active site. Okay, and that active site, in that active site, the suppose this is the enzyme and this is the active site, then the substrate will come to bind with this active site. So what is active site of the enzyme? The site at which the substrate will come to bind. The site of the enzyme at which the substrate will come to bind, that site is called as your active site. Okay, and in that active site, what will occur? There will be occurrence of that reaction, and after completion of the reaction, the enzyme will separate, and the product which are formed here, they will also get separate. So let's see this whole concept in one diagram. Suppose this is your enzyme, and this is normal enzyme that is not yet started its function. Okay, and this is the substrate. Okay, in the enzyme there is presence of a specific site here. Here in the enzyme there is presence of a specific site. This is the specific site at which the enzyme can fit here. Okay, let's see here. The enzyme can easily fit here. Okay, and sorry, this is the substrate. Sorry, this is the substrate. The enzyme and substrate has to be feed with each other. First of all, what happens? This is the normal enzyme or the free enzyme. This is the free substrate. The substrate come to bind with this site, and this is the site of your enzyme, and this site of the enzyme is called your active site. Okay, in the active site, what will happen? In the active site, the substrate will come to bind, and after it bind with enzyme, the enzyme will do its function. Okay, after that completion of that function, there will be formation of your product. So this is one product, this is another product. So this is product 1, this is product 2. Suppose, for example, I am talking here. So after that, this product will dissociate, and again the enzyme is free. Again, this enzyme is free. Again, it will come to bind with that another substrate of the same type. Okay, another substrate will bind with this free enzyme. Again, there will be occurrence of that process of your reaction, and after that, what will happen? Again, there will be separation of your product. Okay, so this is the general things about your enzyme and its substrate and its active site. So what is active site? The side of the enzyme at which the substrate will comes to bind, that is called as your active site of the enzyme. Clear? And after that, here for this temporary period of time, for a very small fraction of time, there will be formation of your enzyme substrate complex, that is called as your enzyme substrate complex. Okay, so this is your enzyme substrate complex, and this is temporary. Okay, the enzyme substrate complex that will form here, and that is temporary, that is for your temporary period of time. After completion of the reaction, again that will get separated, and the enzyme will also get separated, and your product will also get separated. So these are the products, and this is the, again, this is your free enzyme. This is your free enzyme. Okay, let's see the nomenclature of the enzyme. How can we say the name of the, or how can we make the enzyme, how can we give a name to the enzyme? That is the nomenclature of your enzyme. Okay, how the enzymes are named. So according to Duclux, okay, one of the scientists, that is Duclux, according to Duclux, we have to add suffix. We have to add suffix. Which suffix? That is your ASC. We have to use the suffix ASC. We have to use the suffix ASC on the substrate or on the name of the substrate on which the enzyme work. Okay, as you know that there is presence of your substrate, and this is your enzyme. Okay, so we have to give the name according to the name of the substrate. Okay, according to the name of the substrate on which the enzyme work. Okay, जेन substrate उपरे enzyme टा काम करूची से substrate रा ना हिसाब है, we have to give the name of the enzyme, and we have to use ASC. We have to use the ASC just after the name of your substrate. Suppose the enzyme that is acting upon your sucrose, if the enzyme acting upon the sucrose, then that enzyme will call it your sucrase. The substrate is your, here the substrate, here the substrate, that is your sucrose. Okay, when the sucrose will destroy, or when sucrose will degraded, then that is by an enzyme that is called as your sucrase. If the lipid is the substrate, means when the lipid will broken off, when the lipid will broken off, there will be requirement of the enzyme that is called as your lipase. So the here, according to the name of your lipid, according to the name of your substrate, here the substrate is your lipid, so it will become lipase as the substrate is your lipid. And it may be DNA. If the DNA get destroyed, if the DNA get destroyed, then we can get the nucleotides. If DNA get destroyed, we can get the nucleotides. Okay, so for that there will be requirement of your DNase enzyme. So after the DNA, we are using the ASC term, and if we are using the if the proteins are getting destroyed, then the after the name of your protein we are giving the term, or the giving the suffix ASC here. Okay, so this is the nomenclature of the enzyme. Okay, let me check if there is any problem in my networking connection. Okay, I think now it's good. Okay, so just we have to use, according to Duclux, we have to use ASC. Okay, that is used as suffix just after the substrate on which the enzyme will work. Okay, if enzyme is working on the sucrose, then we have to give the name of your enzyme. This is the name of the enzyme, name of enzyme as sucrase. If we are using the lipid as the substrate, then that enzyme will known as your lipase. If we are using the DNA as the substrate, then that will be called as your DNase. If we are using the protein as the substrate, then that will be your protease. Okay, let's see another things. Okay, another things, or in another way we can give the name of the enzyme. according to the source from which the enzyme is extracted. Okay, when we see enzyme are extracted from different plant, different animals. Okay, so from which or from which part or from which organism we are extracting the enzyme. We are extracting the enzyme. According to that, we can use the term, or you can give the name to that enzyme. So one of the name of the enzyme, that is your physin enzyme. That physin enzyme is a milk that is milk coagulation, that is helping in your milk coagulation or milk protein become coagulate. Okay, so milk protein coagulation luggage an enzyme that is your physin enzyme, and that physin enzyme is actually extracted from your ficus, that is your banyan tree. Okay, one kind of your ficus, that is your banyan tree or the peepal tree or dumer tree. These are your ficus. So from the ficus we can extract some of your enzyme, that is called as your physin enzyme. As it is extracted from the ficus, that's why the name is given as the physin. Okay, and some enzyme that are your papain, one of the another enzyme, that is your papain. That papain enzyme is extracted from the papaya plant. Okay, so according to the source from which we are extracting the enzyme, we are giving the name to that enzyme also. Okay, and in case of your biotechnology, we are using one of the enzyme that is called your Taq polymerase. Taq polymerase. Okay, so here the Taq that represent the source from which we have extracted that enzyme. Taq stands for Thermus aquaticus. Thermus aquaticus. The Thermus aquaticus, that is the name of the bacteria from which that Taq polymerase enzyme was extracted. Okay, so according to the source from which we are extracting that enzyme, you can give the name as from its source. Okay, it will just related with the name of the source also. As this is Taq means T here and AQ. Okay, these two term or these three letter that came from the source, and that source is your bacteria here. Okay, so that is your papain, papaya. It is extracted from papaya, that is papain. It is extracted from ficus, then that is your physin. Okay, so according to the source from which the enzyme is extracted, that is another things. Anyway, the most standard nomenclature, or when we see the enzyme are actually divided into 4 different classes. Okay, that is via the International Union of Biochemistry. The International Union of Biochemistry, that society or that union classify the whole total enzyme into 6 classes. Okay, jate bhi enzyme present in our earth or in the nature, all these are come under these 6 classes. So first one, that is a class 1, second one class 2, class 3, class 4, class 5, and class 6. The class 1 enzyme, okay, the class 1 enzyme that are your oxidoreductase, they are performing the reaction, that is oxidation and reduction reaction. If enzyme involved in the oxidation and reduction reaction, then that kind of enzyme will come under class 1. Okay, okay, anyway, here we have to complete this part. Then the transferase enzyme, the class 2 one. Okay, if the enzyme is transforming some functional group, then that is called as your transferase. Hydrolases, if it is just using your water, then lyases. If it is not using the water, isomerase, it will make the isomer. If it is joining some substances, then that is your ligase. So these are the 6 different classes of your enzyme that is done by your IUB, that is International Union of Biochemistry. So in the next class, we will start from this different class of your enzyme. In the next class, we will start from your class 1 enzyme. Okay, thank you.